So it's 7 o'clock now. Welcome, everyone. Thank you so much for attending this evening's AONM webinar on mitochondrial testing with MMD. We're delighted to have Professor Konig with us from Germany. She'll be introducing the tests to you. I'll just introduce her for a moment. She's a professor of medical microbiology and virology, infectious immunology and infectious epidemiology. She has degrees in biology, chemistry and medicine, and a PhD in medical microbiology and infectious immunology. She's written 5 books and 9 book chapters, as well as publishing 130 peer-reviewed articles as first author and 503 peer-reviewed scientific articles. Professor Konig is the recipient of several prestigious awards, including a postdoctoral fellow award from the American Society of Microbiology. So we have a presentation for you this evening, and I'll just introduce a few terms relating to the mitochondria at the beginning. So that'll just be a few minutes, and then Professor Konig, you're there?
I'm here.
Hello, fantastic. And then she will begin talking about the testing, which AONM has just recently begun, introduced to the UK. So next, please, Brigitte. So we'll briefly introduce the mitochondria, then she'll talk about the ATP profile and its various components, then the mitochondrial health index and its markers. And the supplementary biomarkers are quite complex, so we felt that they would probably best belong in a future webinar, which will be very soon. Next. So our mitochondria, as I'm sure you know, are the powerhouses of our cells. They have a bacterial origin. And next, please. It's stunning how many there are in our cells, sometimes 100, sometimes thousands, or even millions in the substantia nigra of the brain where our dopamine is produced. There are actually, or should be, around 2 million per neuron. Next, please. The structure of a mitochondrion is that they have an outer membrane and an inner membrane. And the matrix is the inside of the mitochondrion. They have their own DNA, and cristae are little folds within the mitochondrion. And along these folds, you have 5 little complexes, many, many times over. In fact, there are thousands of these in each mitochondrion. And those are like little football teams that actually produce our energy in what's called an electron transport chain. And the energy that they produce is called ATP, adenosine triphosphate, and that's the energy currency of our cells. Next, please. The cristae are in there to allow efficient use of space. These electron transport chains are really packed in tight. And so that really makes for thousands of them in each mitochondrion. And interestingly, Professor Konig can actually identify each complex in the electron transport chain and work out where there are blockages. It's important to mention perhaps that these are so tiny. There are you could fit about 1 billion on the head of a pin. So that's why it's taken really for electron microscopy and other devices to become accurate enough to identify them. Next, please. So inside a cell, there are many mitochondria, but there's also what's called a cytosol. That's here represented by the blue. And the cytosol can only use glycolysis for making energy, whereas the mitochondria use oxygen. So you have two words there, which will come up in the tests. Aerobic is when you make energy from oxygen, and anaerobic is where you make oxygen, sorry, where you make energy but don't use oxygen, primarily using glucose. So I think there's just one more chart. If you could go on, Brigitte. Thank you. So our mitochondria are responsible for a lot more than making energy, and that's why it's so important to understand how functional they are. I won't go into all these details. I am talking later in the month and will go into this a lot more, but they're responsible not just for our bioenergetics, but also biosynthesis of many, many different cofactors and molecules of different kinds in our body, and also for cell signaling. So over to you, Professor Konig. Look forward to hearing your talk. Thank you.
Yeah, thank you very much, I say Gillian, for the introduction on mitochondria, and welcome to this webinar. As mentioned before, I will talk a little bit about the ATP profile. It's composed of the total ATP a cell can produce, and it's also composed of the capacity to produce ATP by mitochondria or on the glycolytic pathway, and also the capacity to use or to produce ATP by different pathways. The second then is the mitochondrial health index, and it's composed, we will talk about it, about the basal respiration rate, a mitochondrial ATP turnover, a proton leak, a maximum respiration rate, reserve capacity, a non-mitochondrial rate, also oxygen rate, and calculation of the overall mitochondrial health index. And about the supplementary biomarkers, we will talk at another date, because it is a little bit more complicated, and it's what we will talk. We will talk about the ratio of mitochondrial DNA to nuclear DNA. It's a biomarker of how much mitochondria are in the cell. We will talk about a parameter later about PGC1 alpha. It's a biomarker of whether mitochondria are produced, whether new mitochondria are produced. We will talk then also about NERV2. It's very important for the antioxidative response of the cell. And we will also talk about the mitochondrial deletion mutant. It's a common deletion mutant. It is a biomarker for really oxidative stress in a cell, and a lactate pyruvate ratio, which will tell you whether your cell is using glucose or carbohydrates or fatty acids or amino acids, and whether the cell can use the mitochondria or whether it's only producing lactate. So I have a small company. It's called MMD, or Magdeburg Molecular Detections. And we concentrated on the mitochondria and everything what is influencing the mitochondria. So we will always show or look whether we look also other parameters like Epstein-Barr virus or T-cell responses. But the central point, or the central of our work are really the mitochondria. And we use different instruments which are state of the art for the ATP and also for the mitochondrial health index, where we use, you will see later, an extracellular flux analyzer, so that we can do this parameter, or we analyze the mitochondrial functions with a small amount of cells, which was not in former times, and really nearly automatic, so that that mistakes are not, so you are not doing too much mistakes or no mistakes. So I will start with the ATP profile. As mentioned before, the ATP profile is composed of different parameters. First, it is the total ATP. What is the cell producing ATP? And if you remember, if the total ATP, you don't know exactly whether the cell produces it via mitochondria or whether the anaerobic glycolysis, also with lactate formation. So normally the total ATP is one parameter. Yes, it's important, but you need more. You need to know more than only the total ATP. You have to know also how much is the capacity of the mitochondria to add ATP or to produce ATP. And on the other side, you must also know whether the cell is able to degrade glucose and put glucose as pyruvate into the mitochondria. And then you also need whether the cell is able to produce ATP on other ways if there's a need. So we see here a total ATP of 0.8. It is not very good. It is too low. Yeah. And then you see, okay, perhaps the total ATP is produced or can be produced by the mitochondria. And here you see that the mitochondria can only produce 50% of the ATP. And that is really, really low amount. Yeah. So the mitochondria are not anymore able to produce ATP for the cell. So you can assume that the cell needs or produces ATP via lactate formation. But also here you see that the glycolytic ATP is also going down. It's only 63% of the total ATP. That's always referring to the total ATP. So this cell is nearby the exhausting state. And if you look at the reserve ATP capacity, you have 13% of the total ATP. That means that the cell is not anymore able to produce ATP on other ways. So she's really on a bad way. It's exhausted. Yeah, also this way. So you have to do a lot of therapeutic approaches. So that you can, we overgo this. This is what I told 1 minute ago. And the ATP profile is important, sure. It is the first sign to look whether the cell is producing enough ATP, whether a part or the total ATP can be produced by mitochondria or by the degradation of glucose, and whether there's still a reserve capacity for ATP production. But the ATP profile is only able to look at the ATP in cells which are not activated. Yeah? You take the cells and you look what is the ATP profile. But if you only want to know, or you also want to know what is happening when the cell is activated. Yeah? Is it possible to do ATP? Might be that the normal things it's able to produce ATP, but if there is some more need to do something, either by activation by hormones or activation by chemical substances or by infections or whatever, then we want to know, is the cell also possible to respond? And then we use the mitochondrial health index, because here we have not only the ATP profile or the response of a cell in basal rate, but also or in addition to an activation.
Brigitte, can I just mention, you put it under a standardized pressure, don't you? So that it's the equivalent of, as you say, being activated. Like running for a bus, or
oh, that's nice.
You know, a brief period of intense activity. Is that how you could express it? I know it's standardized.
Yes, it is standardized. Sure, sorry. That is sure, you can activate by hydrogen peroxide or whatever, or you can activate it by hormones. No, we really do it standardized. That means we take away the ATP from the cell. Yeah, as the ATP which is produced, we remove, and then the cell has to show what she can do. Yeah, what is her capacity. Yeah, we simply remove ATP. Yeah, what she is just producing. Yeah, so she is producing ATP, we took it away. Then she is sensing that there's not enough ATP. And then she produces more, we take it away again. And so the cell shows us her total capacity to produce ATP under activation.
Understand, thank you.
So the mitochondrial health index is really an index of a lot of parameters. It's not only one parameter. You have to compare or they are interchangeable. So it is the science of mitochondrial health index is very, very old. It's centuries ago, but it was really very well developed at the University of Alabama, which did a lot with the seahorse measurement. That is also the instrument we use because it is standardized and nearly automatic. And if you look, what does a mitochondrial health index mean? I go later on the different parameters which are composed, or which are the parameter, the composition or the composition of the mitochondrial health index. But the mitochondrial health index here, BHI at abbreviation, means if it's very high value, then you have healthy mitochondria. And healthy mitochondria have a high ATP production. They have a high reserve capacity. So if you need ATP, they have a high reserve. They have a very low non-mitochondrial oxygen consumption, so they are healthy, and they have a low proton leak. That means that all the oxygen which is going into the mitochondria is really goes into production, and it's not lost by hydrogen peroxide production and so on. But there might be pathological stresses or whatever, infection, pollution, chemicals, whatever, a pathological stress, and then you get stressed mitochondria. And the stressed mitochondria, you see that they have perhaps a damaged oxidative phosphorylation, so they can't produce too much ATP. Perhaps they have also a high proton leak, so the oxygen is produced into oxygen radicals, which might damage again the proteins, the lipids, and the mitochondrial DNA also. And at the first stage, they also have the stressed mitochondria have also an increased ATP demand. Yeah. They have more ATP demand, but which they can't deliver. And if normally the stressed mitochondria are removed from the body. Yeah, they are bad and they are removed, and new mitochondria, healthy mitochondria, are produced. But this removal of stressed mitochondria is in most diseases or chronic diseases, it's not optimal. So you get a lot of unhealthy mitochondria, also from the stressed to the unhealthy mitochondria. And the unhealthy mitochondria are no longer able to produce enough ATP, but they produce a lot of oxygen radicals. They damage, again, the proteins, lipids. They damage the mitochondrial DNA. And unfortunately, mitochondria with all these damages are producing or are replicating very faster than healthy mitochondria. And at one time, at the beginning, it might be okay, but later you have get a really problem. Yeah. So the key parameters, and I don't go now into the detail, is really a basal respiration rate. That is how much ATP is produced in the cell without energy demand, then how much from this oxygen consumption can be really used into ATP. That is the coupling efficiency, the proton leak. That means is all the oxygen produced into ATP, or do you have loss? Then it's a non-mitochondrial respiration. Respiration is a very important parameter. So what is possible if you have a really high demand? Can the cell respond to that? And the difference between basal respiration and maximum respiration is the reserve respiration capacity. And in addition, there's also an energetic phenotype, which tells you whether the cell is able to use the mitochondria or whether they are using the glycolytic, also lactate formation. And from these parameters here, from these key markers, you calculate the MHI, so it's the mitochondrial health index, which gives you a direction whether your cells are in good condition or in bad condition. So if we look at this mitochondrial health index, it is the summary. You see the mitochondrial health index is 0.77. So that is a very bad value. You see it is marked in red. And the target value, you would like to have it greater than 2.5. So now you see the mitochondrial health index number. Yes, it's nice to have it, but now you want to know what's the problem or where's the problem. So are there a lot of problems or is there a major problem? So if we look at the coupling efficacy, we see, okay, this 100%, that is, yeah, it might be a problem because it's high, but that we see later. So if we say, okay, on the coupling efficacy, we don't look at the moment. We have a reserve respiration capacity. Okay, that's not too bad. But what we see is in red is the non-mitochondrial respiration, 71%. That means that the mitochondria, or that the oxygen which goes into the cell is not produced or not used by the mitochondria, but by other oxidases in the cell. And that means you have a very, very high chronic infection, chronic disease, chronic, you have the oxidases oxidizing metal ions or their chemicals. So you have chronic disease. So then if you look further and you look at the point, the potential maximum oxygen consumption rate in picomole oxygen per minute, you see a very, very bad number. You see 22.99. But the normal range should be greater than 300. That means that the mitochondria is not anymore can't anymore produce ATP, because there's a very low amount of oxygen which goes to the complexes of the mitochondria, the phosphorylation complexes, and it can't be produced any ATP. It's too less. And so you see also that the cellular energy phenotype is addressed, that you have already addressed, you have glycolytic. So the cell is using the anaerobic glycolysis producing lactate. So also in a resting state, the cell is not able to produce enough ATP through the mitochondria. Yeah. So that was, we can summarize that the mitochondrial health index is extremely low, as it's 0.77. And we see, we can calculate from that the mitochondrial dysfunction is extreme. Yeah, we have seen the different markers. So we have very low maximal respiration rate. We have a very high non-mitochondrial respiration or oxygen consumption. So the mitochondrial dysfunction is extreme, and the cellular imbalance is also extreme, because also at the resting state, you have glycolysis, that means lactate generation. And then you look, okay, what are the reasons? It is increased formation of oxygen radicals in the cell. Yes, because we have a lot of non-mitochondrial oxygen consumption, which is used by oxidases, and oxidases produce a lot of radicals. You don't have an oxygen radicals generation at the mitochondria, because we had a 100% coupling efficacy. But we have a restricted function of the electron transport chain in the mitochondria, because the maximal respiration rate was only around 22 and not 300 as we would like. So you have really problems at the electron transport chain. And in consequence, you have also a limited number of intact mitochondria. Yeah? You can't have full functioning mitochondria, then you would have another maximum oxygen consumption rate. So, and you have insufficient ATP formation on energy demand here, because also at the resting state, your mitochondria are not able to produce enough ATP for a resting state. So you have really insufficient ATP formation on energy demand. And yes, we can conclude that you have really an acute inflammation or an active chronic inflammation or autoimmune disease. Yeah? Otherwise, you can't explain these parameters, which in summary give to this very, very bad mitochondrial health. And here's again the main parameters we always look is the coupling efficacy. It was here not too bad that we explained. We have the non-mitochondrial respiration, which is really too high for the oxidases, which are in the cell and which is producing a lot of reactive oxygen radicals. And we have also a very, very extremely low maximum and possible oxygen consumption rate. So we know that we have really bad mitochondria with functioning. And now you are really interested, I think, and okay, if you have some person or some patients with such bad mitochondria, is it possible to cure them? Yeah. First, I say yes, why not? Because mitochondria are derived from bacteria, and they regenerate like bacteria, they divide. So it is possible that you get better mitochondria, also from a low amount to higher amount, yes. But nonetheless, you're interested, is this really possible? And here we show you one patient. It was on 3 different dates. It was in 2020, in October. It was the first one. It was not so bad as we have seen before. It's an example. It was, we have, if you look please at the red, first only at the red sign here with 90.78 is the maximum possible oxygen consumption rate. And I think we will focus at the moment on this parameter because it's a very, very important parameter. So we see that it was low, it was bad. Not as bad as we have seen. It was 2020 picomole. Now it's 90.78. It's better, but it's still bad. So we look at half a year later in May 2021, and then we see it was 123.1. It is much better. And if we look a little bit, only 1 month again later, we see that it's increased further to 180.06. And I think that is a very, very good value. And if you go to the reserve respiration capacity, it's now also increased, sure, to a very green color. So you see at this example that you can cure the mitochondria with therapies. And which therapies are possible, that Gillian will talk about in her next webinar. What are the possibilities that you get this result? But I think the most important message from here is that you can really do something, and that it's getting better. But also you see that you need time. It's not, it depends from where you start. It's not possible to do it in 1 week or with 3 tablets. Yeah, you need really a therapy which may last 1 year or longer. Yeah, sure, you will ask, are our measurements really reliable? I told you that it's also established by different researchers in the US, and there are also many videos available about our seahorse technology, which we are using. And in between, there are over 7,400 studies for which the seahorse has been used. And you see, or you find literature also in Nature, Science, Cell, also very, very famous journals, and also very good studies in these journals about different diseases like diabetes, heart disease, and so on. And on this homepage also from Seahorse, you can also again look at the different energetic pathways, or what they are still really measuring, what they are measuring. And sure, we know that we have a very profound measurement, and that we get a lot of information from our ATP profile and more from our MIH, also Mitochondrial Health Index. But nonetheless, sometimes you need more markers, more information, like a puzzle. And therefore, we developed also supplementary biomarkers. As mentioned before, it's the ratio of, or the number of mitochondria, because it's a very important parameter to see whether your maximum oxygen consumption rate belongs to very high number of mitochondria or very low number of mitochondria. Then you must know, do you have to delete diseased mitochondria, also unhealthy mitochondria, or do you have to increase healthy mitochondria? It's a difference. Therefore, the ratio of mitochondrial DNA to nuclear DNA, or the number of mitochondria, is important to know. Also, it is important to know whether the cell is already the signal is producing new mitochondria by PGG1 alpha. It's also important, if you see that your cell is producing a lot of reactive oxygen radicals, to know whether your cell is already producing NERV2, which is an antioxidant, and is fighting the oxygen radicals. And sure, the lactate pyruvate ratio, to see what kind of foods is using your cell. Is there perhaps a problem in fatty acids use, or in carbohydrate degradation to pyruvate, and so on. And later, sure, we will also go to the more characterization of the mitochondria with further parameters. So the supplementary biomarkers we will talk next time, but they are very important because they are additional puzzle pieces to go more in detail into the right therapy, or to guide the therapy. Yeah. Thanks you very much for your attention. That was a very short one, but just to show you the 2 most important profiles, which you can see, where is your patient? Is he still feeling very well, but you see already that the mitochondria have their problems? Or you can decide do you need 1 year therapy or only 1 week? So this both profiles guide you in your therapy also. Thank you very much, Gillian.
Thank you very much, Professor Konig. That's fascinating. Maybe you'd like to switch to full view so we can see you.
Yeah, sure. Sorry.
Lovely. Thank you very much. Yes, that's, I mean, absolutely fascinating. And I know there's a huge amount of interest in the UK, as probably worldwide, in the mitochondria. It's so hard to get a real handle on what's going on. And the seahorse technology has really opened that up, together with other techniques that you use. One of the questions that a doctor here has asked is, why is the technology named seahorse? Is there a biological significance to the name?
Yes, it's funny. I read it because founder of this technology, the technology is based on the very old Clark electrode. I worked with the Clark electrode since centuries ago, but with the Clark electrode, you need a lot of cells, and with this seahorse technology, you can do it with very small amount of cells, and it's very standardized. That is not with the Clark electrode. Yeah, why seahorse? There are two founders which developed this seahorse, so it was told, and they were thinking how to tell, or which name to give this instrument. And one person loved the sea, and the other person loved the horses. And so they say, okay, then it's a seahorse, and you see the animal, yes, the Seepferdchen in German. And then they say, then they take a combination of sea and horse, and it is a Seepferdchen.
Okay, all right. That's sweet. Yes, just to mention that you do measure you do the analysis on the day that the sample arrives, right? It's very important that the mitochondria are actually alive. And so they do have to reach the lab within 48 hours maximum, which can be a challenge from some countries. As people are asking from various countries, whether Sweden or Hong Kong or whatever, we're checking to see whether it's possible with DHL or FedEx or whatever. But can you tell if the cells are not alive enough, if they don't have enough vitality, and do you then reject the sample?
Yes, we reject this sample because mitochondria, also if a sample, there are two parameters why we can't use them if they are longer than 48 hours. The one is that cells are eating during the transport. They are using the carbohydrates, which is in the serum and which is in the blood.
Oh, that's true. Yeah.
And if they are too long, then there's it depends also on the patients, yeah. But if they have no fatty acids, no glucose, then the mitochondria, yeah, they have full, yeah, and either they will recycled, and so we have a different picture then. A very important point is that during the transport, especially the granulocytes are activated. And if the granulocytes are activated, they influence the immune cells, and that is then a problem. So there's one possibility from Becton Dickinson, which that you can transport the cells up to 96 hours, but then they must be centrifuged in a specific medium before the transport.
Oh, well, we'll have to find out about that because we're not using that yet.
Yes, but you have to have a centrifuge to do it. And the most doctors don't have this centrifuge.
Most clinics in the UK that are drawing blood do actually. So that's something we can discuss.
Okay.
Yeah, that
so
would require different tubes, presumably, not the CPDA
yes.
we have.
Well, yes. So this is two parameters. And we sure we did validation, and perhaps some people like to know it, I don't know it, But sure, we also looked what is happening with the blood if you transport it more than 48 hours. Yeah, so at once, immediately 24 hours, 48 hours, 72 hours, 96 hours, and so we did it. And it depends a little bit on the patients. If you have, by some patients or some people, also healthy people, then the mitochondria are getting better after 96 hours.
Oh, wow.
Yes, it's simple because the autophagy is functioning. They don't have enough food, and then they they delete the mitochondria which are not anymore functioning.
Ah, that's autophagy. I understand.
Yes, autophagy.
Yeah.
Yeah. So it's functioning, and then you have after 4 days, you have only the good mitochondria. Yeah. And another part of patients or people, they're getting worse. You see no anymore mitochondria, yeah, because the autophagy is not functioning, yeah. And then you get worse and worse and worse, yeah. But 48 hours, it's not a problem, yes, with our CPDA tubes, because there's inside glucose, adenine, so they have enough food, yeah, for 48 hours. And the granulocytes are not activated, but after that, you see either you're getting better or it's getting worse.
It's a standardized process. Yes, we have specific monovets, and the CPDAs are specific to the mitochondria. It's not possible to take the CPDAs from, for example, the AONM Labs tests and use those. These are very specific ones for the terribly delicate mitochondria, and they have, as you say, been through. So just to also mention a question here, which is very interesting. Might oxygen therapy be beneficial to upregulate the number of healthy mitochondria? Can raw electrons in oils, seeds, or vegetables actually help?
Also the oxygen therapy, there are different oxygen therapies. Now it is important to know which type. Hyperbaric oxygen therapy, ozone therapy, or intermittent hypoxia or hyperoxia, different. So every type of oxygen therapy has its good sides, but we have to know which type of oxygen therapy, and it's not always. It depends on your mitochondria. Sometimes it's better first to cure them a little bit before you start with the oxygen therapy. Yeah, it depends which type. We did some analysis. If you have very bad mitochondria, then it's not good to start with the hypoxia or hyperoxia, because then you need also functioning mitochondria that you have the benefit, and if you don't have it, you have no benefit, or it's getting worse. So it's very, you can use this test to guide which type of oxygen therapy and in which, how much, how strong, and so on. Yes.
Oh, that's fascinating. And can you actually tell from the results of these tests that we've discussed today whether a patient has an infection ticking away in their cells? Can you be sure that it's an infection rather than heavy metals or some other problem, like the plastics you talked about, or the different types of pesticides? Can one actually tell that, or does one need to do other tests alongside?
Ideally, it's better to do some additional tests to really look where's the problem. Sure, you can get some direction. Yeah, it depends. Your lactate pyruvate ratio tells you a little bit more whether it's an infection, in addition to the non-mitochondrial oxygen consumption. So you get something, but it should be better to confirm it with other tests.
Thank you. And the lactate pyruvate test that you do, just because you've mentioned that a few times now, that can, I believe, show whether the patient is primarily using glucose or fats to feed their mitochondria. I mean, should fats be the primary fuel source for mitochondria?
It depends. It depends on the type of cells you are talking about. We, at the moment, we use now for this test immune cells. And
they're PBMCs are
Yes,
the
peripheral blood mononuclear cells we use. And peripheral blood mononuclear cells are composed also on different cells. So you have some memory T cells, you remember, which remember the infection. They more rely on fatty acids, very, very strong, while macrophages go more half and half, but you have only a few percent of macrophages in the blood. It depends a little bit, but if you use the peripheral blood mononuclear cells as PBMC, you can say it should be mainly fatty acids, also should be a little bit more than glucose, yeah, in this direction, yes.
And are there ways, if you see that, for example, complex 1 is blocked and it's difficult to get the nutrients into the mitochondria, are there ways of leapfrogging that, jumping over that and getting the fatty acids into the mitochondria anyway?
Yes, yes, we test individual complexes, and so we give them everything they need. So we see whether there's a problem with the complex by itself. Yes, we do also one test where we look whether ketone bodies are better. Yeah, so it's not the oxidation. Otherwise, if you have fatty acids, you must oxidize them. First, you have to degrade them in the oxidases, also in the peroxisomes. And then you have also sometimes a lot of oxygen radicals. And then we can look whether ketone are at the moment useful to use them. Yeah. Mm.
Just to mention, by the way, that we've said that it's important to please get the bloods to Germany within 2 days. That means doing the tests ideally on a Monday or a Tuesday. In some parts of the UK, Wednesday is fine as well if there are a lot of pickups in that area. And AONM will help to ensure that the courier goes smoothly. They provide the waybill for you. The tubes that are supplied have an adapter with them. The needle has an adapter with a special extension tube that makes it easier to draw the blood, because we've found that quite a few phlebotomists are used to using vacutainers and not monovets. But in fact, the NHS hugely on monovets and has no problem drawing blood with those, but these adapters have made it very easy. Do you find that the test results, by the way, are quite extensive? The ATP profile is not just what you've seen today, it's a 5-page set of results, so quite a lot of explanation. And the MHI, the Mitochondrial Health Index, is 11 to 13 pages, depending on whether you're repeating the results or not at the end. But how quickly would you say, what's the turnaround time for these tests? I know that at the moment we need a little longer because of going over the English and smoothing out things, but in general, you've got the results quite quickly, haven't you, because of the swift turnaround that's necessary?
Yes, at the moment you're right. Because we are looking to do it better, to nicer, to the English writing and so on, the spelling. Normally, also as if the blood arrives, the test is done. So the test results are available on the same day.
My goodness. Okay, so if it's essential, we can sort of call you and get the results. Yeah.
So we have we also look to optimize that so that somebody can do it on the next morning to put it in this profile, see, and then it should be ready. That is optimal, and we are working on it so that we have enough people, because the result is ready really on the same day, and so that is put into this profile or into this reporting the next morning.
The interpretations obviously take a little while, because the interpretation isn't the same for every patient, obviously. Yeah. Do you find that it helps to do several tests? Do the additional supplementary markers and also doing the ATP profile as well as the MHI, can one get a lot more information from this?
Yes, yes. Ideally, I like all because they are really puzzle pieces. And you see, if you take all, you have a really full picture. Yeah, sure, you can add exactly noch again how the individual complexes. Yes, you can do it. But if you have this, the ATP profile, the MHI, so, and the supplementary markers, you have really full picture.
Yeah. I mean, I find it very interesting that the ATP profile is the cell at rest.
Yes.
And then the MHI is when it's put under pressure. Do you often see quite a big difference there, that the person's cell at rest seems to be functioning quite well as far as the mitochondria are concerned, but the moment they put themselves under pressure, this would be what's called,
Yes.
you know, the post-exertional malaise.
Yes.
That's how we term it, you know, that the person really needs to rest sometimes for a full 3 days until they've remanufactured their ATP. It's like de novo synthesis.
Yes.
You know, they really, really notice even just a small amount of energy utilization.
Yeah.
Do you sometimes see a big difference between the two?
Yes. We see either that both parameters are okay, so they're really healthy. Then we see that the ATP profile is looking nicely, but you have already a deficit in the MHI because the reserve capacity is not optimal. And then the next stage is really that already at rest, you have a very bad ATP profile, and in addition also then at energy demand. Yes.
Okay, thank you.
So you have never the profile that you have a bad ATP profile and a good MHI. That's not possible.
That's impossible.
No.
Thank you. I know that it's possible for you. I mean, obviously that's a further stage of both, you know, work, thinking and so on, but you are able to provide some recommendations as to how to improve the function of the mitochondria. I know a little bit like with our OAT tests here, that's the organic acid test.
Yeah.
They will provide a sort of an entire list of nutrients and how much to up them by as the front of the test. I know you can do that, but it's sometimes a little bit deceptive, isn't it? Because if the patient does have an infection or heavy metals or contaminants in their cells that first need to be removed, or if they're under heavy stresses of different kinds, even EMFs, you know, EMF stress or consuming the wrong kinds of foods, then just looking at those the pure mitochondrial nutrients can be making it a little bit too easy, let's put it that way. But I know that you are able to do that. So that's a further stage that we will get to in the future, where we can make those suggestions if people wish to have them. But just to mention that while Brigitte, you kindly mentioned that I would be speaking on the 26th of April about, it's entitled Mitochondrial Magic, Tips for Revitalizing Your Mitochondrial Health. So I look forward to that. I'm absolutely passionate about the mitochondria, and I hope you'll perhaps take part in that as well. It'd be lovely. But on the 12th of May, we're delighted to have Dr. Sarah Myhill, and that's actually International ME Day. So it's a very, very special day. And she is sort of the mitochondrial queen of the UK. She's done so much work on the mitochondria, and she'll be speaking about mitochondrial dysfunction and health as well on that day. So we're delighted that she's able to make that time. I think it'll be 6 o'clock rather than 7 o'clock, but we'll be sending out information. So I was fascinated to notice too that recently many seahorse tests have been done of SARS-CoV-2. You know, most of the studies actually, if you read the studies of COVID-19 or SARS-CoV-2, whichever term you use, seem to be done with that technology. I think because it's so clear that it is driving glycolysis in cells, isn't it? Yeah. I mean, probably like most viruses and pathogens, but it's really utilizing the glycolytic pathways, and you can see that so clearly. Do you do tests of that kind as well? Do you see a difference, for example, between when someone has had COVID and then when recovered or when they are recovering? Have you done tests to see that difference? I mean, I think it's called long COVID.
Yeah,
unfortunately. Germany as well.
Unfortunately, not. We have some tests done on patients with long COVID, yes, but I don't have the values before the SARS-CoV-2 infection. So exactly whether they had bad mitochondria, so they get long COVID. Yeah, that might be one reason. But the cells, the mitochondria we measured, they are terribly bad.
Terribly bad in long COVID if a person is really suffering from it.
Yes.
Oh, yeah. And have you got an idea of, you mentioned how long it can take for people to recover. Can you tell from the tests of this kind that we've looked at today, approximately how long it will take for the patient to recover if they're given therapy? Or is it very, very variable?
Sure, it's variable, but you can give this advice because also in addition, if you have the supplementary markers, normally if you have a cell and if she doesn't have enough ATP or energy, then the cell starts to produce more mitochondria to get more ATP. And then you see that if you then do the supplementary marker with the mitochondrial DNA to nuclear DNA, also the number of mitochondria, you see that you have an increase. Yeah, you have a normal ATP profile, but you have an increase in mitochondria. And if it takes too long, then the cell, because she has a lot of mitochondria, needs again more ATP, more energy, and then you go to the reverse, you go to low amount of mitochondria. And you see also whether the PGC1 alpha is, also this signal to produce more mitochondria is low or is high. And you from the normal MHI profile, you also see whether the reserve capacity is very bad, you need more time. Yeah, so you can assume from that where it is, whether it's perhaps just 1 week, yeah, or just perhaps vitamin B12 or whatever, it's very one substance, or whether there's really a lot of disbalances so that you need really a lot of time. Yes,
a lot of dysfunction.
Yes.
Will you be prepared, at least initially, to look at the tests individually, or perhaps even putting together groups of therapists if necessary to help them to analyze the tests?
Yes, sure.
Oh,
that I do also in Germany. Yes, but then they also provide me with additional parameters because I don't know the patient. And so it's helpfully if they give me addition, or if I ask them and then they give me the information, and then we can profile in the context. Yeah.
Oh, that's super to know. Thank you. Okay, well, I don't want to take us a lot after 8 o'clock UK time, because I know it's an hour later in Germany. But do you think that maybe in 6 weeks or 8 weeks we can go through the additional parameters?
Yes, I think
Supplementary ones? Yes, that would be lovely. Thank you. And we'll see you again, hopefully on the 26th of April. That's at 7 o'clock.
Okay.
And, you know, let's do that together. It would be lovely to have you there as well. And I'll give my tips and you can add, you know, information that you have from any tests. Perhaps we'll introduce a couple of additional ones that we have. And then on the 12th of May, 6 o'clock, we'll have Dr. Sarah Myhill. Please be present, you know, for that one as well, if you can. And then we will, as promised the last time when we had Dr. Schwarzbach talking about the way in which SARS-CoV-2 has reactivated viruses. We were going to have a webinar where we talk about therapies that are available for those kinds of viruses, especially EBV and varicella zoster virus and the enteroviruses. So we will be preparing that and offering that very soon afterwards. we did have a wonderful conference just over a week ago with Biolab on for long COVID, and that will be available from Biolab to purchase if you weren't able to take part. I think it would be very worthwhile. I mean, forget about mine, but so many wonderful other therapists and doctors were there, and there were lots and lots of lots of advice given. So and Dr. Rachel Nicol gave absolutely superb information about long COVID from many different perspectives. So thank you so much, Professor Konig, for spending the time with us this evening, and thank you everybody for being there. We will be putting up the recording in the next few days on the website and sending around notification that it's available. And so look forward to seeing you again, and thank you very, very much.
Yeah, good evening to all.
Thank you. Bye-bye then. Bye-bye. See you all again soon.